This is one of the first in-person lectures that we're beginning to have, not just for you guys, also for him and for me. So the world is beginning to just slowly turn again, just beginning to, if you noticed. And what we want to do today is take advantage of a very special person, Robin here, our guest, who is recently transitioned from Imperial College in London, where he was since 2008 with a short stint at Oxford, to UCSF, where he's the director of a new psychedelic research program. He's going there as a distinguished professor, and don't let his youthful and sort of soft-spoken ways distract you from the fact that he's one of the leading and foremost authorities on psychedelic science and psychedelic research in the world, not just in North America. Robin is here when basically he has not even unpacked his luggage over at San Francisco. He's just taking a few days to settle down and checking out Disney and checking out the Brain Institute over here. He was one of our first guests at the Brain Institute when we just opened, even before we opened, sort of when we opened some years ago, maybe 3 years ago, just about. Back then, I went to San Diego with Alice here, and we kidnapped him for a few hours. We showed him around, and then we drove him back. He recovered partially from that experience, and maybe psychedelic drugs help with that. We're going to hear about that today. But psychedelic drugs have a lot to do clinically with not just mental health and resilience and helping particular psychopathologies, but also with human consciousness, understanding, self-development, things of that nature. And part of the many contributions that Robin and his team of researchers are doing is trying to unlock and unravel the science of psychedelics vis-a-vis brain mechanisms, vis-a-vis clinical applications. And that's what we want to hear from him a little bit about today. There's a limit to what he can say in an hour, and we'll just we'll take what we can get. Now, I realize that some of the people here have like more of a pharmacy background and so on. Feel free. This gentleman has a background in psychopharmacology, as well as training in psychoanalysis and psychosocial science. So I would want to just take this opportunity to tell you how excited we are to have Robin here, and we look forward to having a long and fruitful collaboration while he's in San Francisco. I expect Robin to continue to move and shake the field of psychedelic science in a big way, and a warm welcome to Robin. Thank you for joining us.
Thank you, Amir. Thanks, Amir. This is actually the first talk that I'll be giving in person since COVID happened. So I don't know how many in-person talks you've been to recently, but this is my first one and I'm delivering the first one. So it's going to be interesting for me. And just a word of appreciation to Amir for having me over again. When I visited something like 3 years ago, I was made to feel very welcome then, and the same again this time. And just when Amir was introducing me to some of his colleagues earlier today, I was really impressed how you know my bio so well. Most people, they get sent the bio and they read it word for word, and then you feel kind of embarrassed that you wrote that thing in the first place, But Amir knew my A levels, which are the subjects that you pick at the tail end of high school before you go on to do your first degree. And I was really impressed with that. Yeah, so thank you. So this, in a sense, speaking to the point that you made with my A levels, which were chemistry, sociology, and psychology, of kind of eclectic interests and not necessarily subjects that you would associate with each other. In a sense, this talk will, speak to that kind of ethos a bit. It's multi-level. Psychedelic drugs is the common denominator. Was it there when I chose my A levels? I don't know. We'll move on from that. But they are, it's kind of somewhat, unique's a dangerous word to use, that one, whether there are, other examples, but psychedelics seem to cover so many different domains of study of subject matter, from art, philosophy, music and culture, sociology, all the way through to, hard science, chemistry, perhaps even principles of physics. So they are kind of unusual in that respect. And you'll pick up some of that multidisciplinary appeal in this talk, I hope. I'm learning these days that it's right and proper to get disclosures in, and I'm grateful that they even exist. In fact, one of them is responsible for my current position now at UCSF. The founders of Compass Pathways put up the funds that created that position for me. So disclosures there. So we begin in terms of the science at the most, really the most salient, lowest level, the molecular or proteomic level, which is the receptors in the brain that psychedelics first bind to and stimulate. They're agonists, and they're agonists at a certain serotonin receptor subtype. Yes, they're non-selective. They'll bind to other receptors in the brain, but we know that this particular receptor subtype is very important, critically important, for a few reasons. First of all, it's important to tell you where the receptor is in the brain. Here's a PET image using an agonist ligand. So this is a radioactively labeled serotonin 2A receptor, selective serotonin 2A receptor agonist. It's labeling the receptor in its functionally available or active state, its high affinity state. And so this is the most, in a sense, informative map in the living brain, the living human brain, that we have for addressing this question of where is the serotonin 2A receptor in the brain that we have to date, and it's relatively recent data. And the important points to emphasize are that it is very much a cortical receptor. You can see relatively little expression in subcortical regions like the thalamus there and medial temporal regions, but high expression in the cortex, and especially in high-level association cortex or transmodal cortex, crossing different modalities, different modules. And that's important. We'll come back to that. So that's where the receptor is, hierarchically speaking. It's high up. It's at the high end of the anatomical and functional hierarchy of the brain. We know that the receptor is very important for a few reasons, as I said, and really the first evidence for its importance came in the mid to late 1980s, I think it was, when it was demonstrated that the affinity of a range of different psychedelic compounds correlates with their potency. And the rule is the higher the affinity, or the higher the binding potential, or stickiness, if you want, for this receptor specifically, the higher the potency. So for example, LSD has a very high affinity for the serotonin 2A receptor, and it is very potent. It's psychoactive in microgram doses. We're starting actually through interest in microdosing, the use of very, very low doses of psychedelics to get a sense of what the threshold dosage for activity might be. Broadly speaking, around about 15 micrograms. Remember, there's 1,000 of those in a milligram. So if we think of another psychedelic compound, psilocybin, a compound that occurs naturally in certain species, certain genus of mushrooms, the psilocybe genus, magic mushrooms, they were first referred to in the late 1950s, I think it was. That is active in milligram doses. The typical oral dose of pure psilocybin is, in terms of the therapeutic work, around about 20-25 milligrams of psilocybin. So, you know, an order of magnitude less potent, and that is mirrored by its relatively lower affinity for the 2A receptor. Let's drop down another level to mescaline, which is a further order of magnitude lower affinity and less potent. So Aldous Huxley, writing his famous essay on his mescaline experience, given 400 milligrams, close to half a gram of mescaline, given to him by Humphrey Osmond, a British psychiatrist working in Canada. And it was Humphrey Osmond, in conversation actually with Aldous Huxley, that coined the term psychedelic, meaning to reveal or to make manifest the psyche. So a few tangents there, but the important point is that mescaline, lower affinity for 2A, still a 2A agonist, still a classic psychedelic, but much less potent, hence the dosage required for it to have its signature subjective effects. We also know of the importance of the 2A receptor through antagonists, so blocking agents, antagonist pre-treatment studies, where if an antagonist is given before a psychedelic, whether in non-human animals or in humans with typically ketanserin, non-selective, but still some selectivity for the serotonin 2A receptor, then you give the psychedelic. Effectively, simply speaking, the signature psychological effects of the psychedelic will be abolished. They'll be muted by that pre-treatment with a blocker. The blockers got in the way of stimulating that receptor. Now the agonist comes in, it can't hit that receptor, so you don't trip. So this is quite compelling evidence for the critical importance of the 2A receptor. We also now have more recent work looking at percentage occupancy of the serotonin 2A receptor by given dosages in this study, PET study, displacing a hot ligand, this SIMB 36, I think it is, highly selective serotonin 2A receptor agonist. It's stuck there on the receptors. Then you give psilocybin and you displace that hot ligand, and you can work out the percentage available 2A receptors that are occupied by your given dosage of psilocybin. It's a small study because PET studies always are. It's a very expensive modality. But here the authors have looked at a plot relating the subjective intensity to just subjective ratings of intensity of effects with the percentage occupancy of serotonin 2A receptors. And what we can see is that it's not written on here, but these are different dosages of psilocybin in a small number of different participants. And the dosage that achieves around about 60% plus occupancy is around 20 plus milligrams of psilocybin. You're getting strong subjective effects. And this is the percentage of receptors occupied. That's just really confirmatory evidence, again, for the importance of the receptor. The more that you're occupying with drug, the bigger the experience. That's kind of obvious, but still to demonstrate it is important. As suggestions, it's a small sample, it's too early to say, but they plotted this in a nonlinear way. So maybe there's some nonlinear relationship between the occupancy of 2A receptors and potency. Perhaps above a certain level, you get a kind of inflection, it jumps up, and so the experience jumps up in intensity. It takes on a different quality and intensity. So that's important for just laying the foundations, the basic pharmacology of classic psychedelics. If we had to define these compounds by their phenomenology, it would be a challenge. And I think we would have a vague definition and opinions would differ. So I quite like the harder pharmacological definition of classic psychedelics, that they share this property of being direct agonists at the serotonin 2A receptor. Those are classic psychedelics. LSD, one could argue whether that's the prototypical psychedelic. It was after LSD was discovered in its psychoactivity, at least in the early 1940s by Albert Hoffman, that there was really in a period of intensive research with psychedelics into the 1950s and 60s. So one could argue it's the prototypical psychedelic. The term psychedelic was coined in 1956. although one could also argue that maybe it's mescaline, given that Huxley and Osmond were discussing mescaline when the term was first coined. But anyway, LSD, psilocybin in magic mushrooms, DMT, you'll hear about that later, an endogenous psychedelic, albeit in very small trace amounts, and mescaline as well. Those are some classic psychedelics, direct serotonin 2A receptor agonists. So if we know that this receptor is very important, critically important, then a natural next question is, what does it do? What's its function? Why is it there? And the summary property is really plasticity. And so most broadly defined, a dictionary definition of plasticity is a phenomenon, a system that can be more easily, it's the property of being or easily shaped or molded, or to change that phenomenon. So that's plasticity in the broadest sense. And usually when we refer to plasticity, especially in neuroscience, of course, we're referring to neuroplasticity. A range of different markers of neuroplasticity have been found to increase with administration of psychedelics. Recent work published in Neuron by David Olson's lab, UC Davis, looking at different components of synaptogenesis and finding quite marked increases. Yes, this is a schematic, but making the point that these different psychedelics that were looked at all quite markedly increased synaptogenesis post-exposure. But this is more recent work on top of older work looking at BDNF as a marker of neuroplasticity, a near doubling of that in the cortex specifically. Here in the dentate gyrus, it's actually a decrease. So in line with what we know about the location of the serotonin 2A receptor, that it's very much a cortical receptor, it sort of backs this up, that it's very much a rule associated with cortex more than subcortical regions, an increasing cortical plasticity, and now a number of studies showing that. Now we've known for a while that serotonin is very much involved in brain development. But now there's increasing evidence to link that specifically to the serotonin 2A receptor itself. And perhaps the most exciting finding in that respect is this one published again in Neuron in 2020 that looked at basal progenitor cells and their proliferation. And they found that there was a seemingly quite selective relationship between the 2A receptor signaling and the control of the proliferation of basal progenitor cells, pre-neurons, if you want, or neural stem cells. And they were markedly increased in a species-specific way in human embryonic brain. That wasn't being seen in other species. So this very much excited this research team that already have a track record. It's Wieland Hüttner's lab at Max Planck, have a track record of doing research specifically related to brain development and brain evolution. And they make the point in the abstract that this potentially suggests a role for serotonin 2A receptor signaling in cortical expansion, put it another way, in brain expansion, in brain evolution, the evolution of the human brain that has such, as we know, such a large cortex. And so quite a remarkable finding, if that's true, that the receptor that psychedelics hijack in the adult brain, when people are administered psychedelics or they take them, early on in brain development is associated with brain growth itself and potentially brain evolution. So it adds a certain, intrigue and context to this receptor. And, more work to do there, but for me, it sort of fleshes out an understanding of the function of this particular receptor. We could also look at learning, and this is low-level learning, associative learning, and how that certain aspects of low-level learning, at least learning rate, can be accelerated with exposure to psychedelics. There's a lot of work in rabbits looking at this. This is aversive learning, air puffs, and an acceleration of associative learning with LSD, and it being slowed down with pre-treatment with a 2A blocker here. We've got some more modern data which supports this basic finding of an accelerated learning rate with 2A agonism. Now, this isn't going to translate necessarily into learning complex information, but in terms of low level, perhaps implicit learning, some pro-learning effects with psychedelics. Also, extinction learning has been shown to be enhanced with psychedelics. And as we'll go on to later on, this potential therapeutic application of psychedelics in terms of unlearning, if you want. Think of addictions, habits that become entrenched, stamped in. Then if we can increase extinction learning, the ability to unlearn certain unhelpful associations, then that may well speak to the therapeutic potential here. We can jump up a level of abstraction to look at cognitive and psychological flexibility, and also evidence of increases in those different domains with exposure to psychedelics. This is a rule that seems to hold up most strongly post-acutely or subacutely. So not necessarily during the acute effects themselves, but afterwards there's evidence of increases in the ability to set shift, for example, reduced perseveration, and the ability to sit with certain aversive memories and emotions, cognitive flexibility associated with certain schools of psychotherapy like acceptance and commitment therapy. So the ability to sit with discomfort rather than seek avoidant defense mechanisms, avoidance strategies, for example, useful, of course, in the context of psychotherapy. So there we have it. There's really the take-home point with that slide. We started with the 2A serotonin 2A receptor, critically important for the action of psychedelics. You find it in the cortex, you find it in the high-level cortex. It's involved in plasticity, 2A signaling, increased cortical plasticity. So then you ask a question that's sort of another sort of natural, logical next question, which is under what conditions, in a sense, forget about psychedelics for a moment, under what conditions might you naturally engage this system? And so over the years, I picked up that conditions of adversity seem to be associated with an upregulation of this system. So in a recent review paper, which has a title, Pivotal Mental States, we summarized some of these evidences. And what came through is that a range of different stressors, here we've got hypoxia, but we've also got chronic stress. I think it was electroshock, we have separation, and chronic immobilization stress, upregulating the 2A receptor, whether it's the sheer availability of receptors or their sensitivity to signaling here in these two studies. But a range of different stressors, and particularly it seems chronic stress, repeated stress, seems to prime this 2A system. It sensitizes it and it upregulates even the availability of the receptors. So if you add to that kind of context, acute stress punishment paradigms seem to be the most reliable way to release serotonin in the brain. Here we're looking at tail pinch, handling, and swim stress, or big spikes in synaptic serotonin release. But you've got this kind of perfect storm of a background of adversity, and then on top of that, some acute stress. These are the, in a sense, natural or drug-free conditions under which you will engage this system. Psychedelic drugs are just a drug way, pharmacological way of hijacking this natural system. And again, I show this and I think about this because it's about contextualizing these compounds and what they do in order to better understand them. And it helps me, I hope it helps others, but one of the ways in which it helps me is to think about, in a sense, the outcome agnosticism of psychedelics, and also the importance that we put on the context in which psychedelics are used in terms of the outcomes that are observed. So, for example, when we do psychedelic therapy, and the clue's in the name, this isn't just a drug, it's a combination treatment, in a sense, a drug-assisted psychotherapy. We're engineering the context in a particular way. We're trying to make it conducive to therapeutic development. If you don't do that, if you just give a potent mind shaker, a potent hyperplasticity drug, and the conditions are adverse, then you might have conditions that would be conducive to a worsening of, for example, someone's mental health. And so in psychedelic therapy, we're trying to engineer things in a particular way, a positive way, a nurturing way. And yet sometimes people take psychedelics, or they're given psychedelics and aren't even told the situation isn't right for a psychedelic experience. Perhaps there's some personal vulnerabilities, and there are these rare cases of what you might call iatrogenesis, or a worsening of mental health presentation in such contexts. Of course, we don't want to see those things happen. They do happen. It's a shame. It's an abuse, in a sense, of these potent compounds, and perhaps a more, well, not even with the perhaps, a more, responsible way to use these powerful drugs is in a therapeutic way, with informed consent and the right therapeutic container. We can induce states of hyperplasticity and create a context that's supportive and nurturing, and with that combination, help pivot someone towards improved mental health outcomes. And really, although it may be a complicated way of saying something quite simple, of just harnessing this plasticity, this ability to change, and then trying to shepherd it in a particular direction. That's the model. That's psychedelic therapy. And the term pivotal mental states is really just referring to this opportunity, this plasticity, induced plasticity, and then an opportunity for change. It's a pivotal opportunity. One could pivot towards wellness, but in less positive context, there could be a pivot towards psychopathology even potentially. So that's the model there. Outcome agnosticism is how I refer to it. Now, that kind of sets the scene for thinking of the modern research with psychedelics. And there are some important take-home points here. In a sense, this material is quite dense and could easily occupy an entire talk on its own. So I just want to emphasize the key take-home messages. The first one, and they're highlighted in red here, is that in all of these studies that have looked at a range of different domains of mental health, there has been an engineering of the contextual conditions. Historically, people have referred to context. They've unpacked it into two particular dimensions: set, which is mindset, and setting, which is the environmental context. But all of the researchers that have done these modern human studies, really starting in terms of the clinical research in 2006, Roland Griffith's work and Francesco Moreno down at San Diego, I think, looking at psychedelics and mental health outcomes, but they've all engineered the context in this positive way. I think across the board, all have featured music listening. That's a detail that if you didn't know this space, it would be easy to overlook. You might even, hear of the non-directive nature of psychedelic therapy, eye shades, people lie back on a couch, not much is said for many hours. And yet people are often transformed in that scenario. And so you might think, well, this isn't really psychotherapy. This isn't talking therapy at all. Where's the talking? But, you know, there is what we've referred to in the literature, a kind of hidden therapist. We've used that term in relation to the music. So the direction is subtle. It's harnessing music and its power to evoke, to direct people as the sort of main directive influence in the sessions. So that's one take-home point. The other one is that if we look here, we're looking at treatment-resistant depression, depression severity scores dropping 1 week post-treatment. So there was a lot of excitement generated by ketamine when it came on the scene as a rapid-acting antidepressant. So this is just to make the point that the same is true if you look at classic psychedelics, rapid improvements in symptomatology. And also, and this is a potential advantage versus ketamine, the improvements appear to be more enduring. Why is that is a fascinating question. There's some recent work, I think it's buried in here somewhere, looking at plasticity markers and more enduring increases in synaptogenesis. Very recent work, more enduring increases in synaptogenesis with classic psychedelics, and I think specifically psilocybin. In the same study, they looked at ketamine and they were more shorter lived. So whether we can put it all down to synaptogenesis in terms of explaining that, or whether, you know, you can appeal to both a brain change and a psychological change. People often, if asked, you know, what is it that's different now? Why are you well now? They'll often refer to insight, psychological insight, personal insight about the nature of themselves, their lives, their suffering, how it came to be. And so there's this epistemic transformation. They learn something, they know something now. And that seems to be related to why they stay well. And that was very common in our depression trial, that people would describe it that way. And now I look at things a different way. I have a new perspective on things, on my depression, on how I went into myself after I lost my mother, or whatever it might be. So the rapid and enduring, also this transdiagnostic action. You can see a lot of different indications listed here. We're doing a study in anorexia now. It's early days, but it's going very well. Psilocybin therapy for anorexia. Chronic pain is being looked at. Addictions, there's some literature on tobacco addiction, Matt Johnson, alcohol dependence as well. But now there's cocaine addiction, Peter Hendricks at Birmingham. There's, other addictions being looked at. Broad range of symptom domains, diagnostic categories. People are looking at psychedelics. So you might just think, are people getting carried away with all of this? Or maybe there is some, you know, method to the madness. Maybe there is a common denominator that psychedelic therapy can hit. And I think if that's the case, then it does invite a question of what's a common denominator to these different psychiatric disorders or symptom clusters. And others will have said this, but the way I articulate it is in terms of overweighted, reinforced ways of thinking and or behaving. Whether it's, say, in an eating disorder where you see yourself a particular way, maybe that's entirely at odds with how others see you, but you think you're too big and so on. So there's these biases that get reinforced, that get stamped in and then dominate your thinking and behaving in a pathological way. They become overweighted. These models of mind and behavior become overweighted. So in that context, you know, a effective treatment and effective intervention might target the weighting of one's assumptions. And that might be your window of opportunity for combining that with psychotherapy to try and promote a positive change there. And that's how I suggest psychedelics are working, that they come in and they open a window of plasticity, potential change, and then combine that with the right kind of therapeutic container, right kind of support that can work towards a healthy revision of these pathological ways of thinking and or behaving. And that's what I propose is, you know, part of the mechanism, the transdiagnostic therapeutic potential of psychedelic therapy. So this is now shifting focus onto some of the brain imaging work that has been done with psychedelics. Here we're looking at psilocybin and looking at communication patterns across different regions in the brain, dots on the periphery here. And the different colors are different communities of brain regions. You can think of them as brain networks or systems. And the point to make is that ordinarily the communication is strong within certain systems, networks, modules, and relatively weak across modules. And that pattern shifts very dramatically under psychedelics. Here, psilocybin, much more communication going on between different modules in the brain. And we've looked at this basic quality of brain function in a number of different ways now, here with a different drug, LSD, but also rather than looking at sort of whole brain connectivity, focusing on a region of interest, the primary visual cortex identified with retinotopic mapping, and then asking where in the brain is spontaneous activity coupled over time with that seed region. Ordinarily, it's regions, other regions of the visual system where activity is synchronous. That would stand to reason. But under LSD, it's most of the brain is functionally coupled with V1. Now, this invites ideas, and actually we saw a relationship between the complex imagery that people would report, but also rate under, or rather after LSD, and the greater the magnitude, the intensity of complex imagery, the greater this increase in whole brain communication with V1. So it invites ideas of synesthesia or inter-module mixing and influence, other domains starting to, in a sense, bleed their function into otherwise quite a specific functional module, the visual system. And another drug, DMT, here we give DMT intravenously. And we're looking at global functional connectivity here. The hot colors are regions whose activity has become significantly more coupled to the rest of the brain, the average of the rest of the brain. The pattern, you'll see it again in a moment, overlaps with the spatial location of the serotonin 2A receptors themselves. High-level cortex, associative cortex, transmodal cortex, it seems to be being particularly, sensitive, particularly hit hard by the psychedelic here. And in a sense, its modularity is breaking down and it's becoming more globally coherent in its activity. And this is one way that we've looked at that. It's a more dynamic analysis. These are different brain modes or states. There's no real reason to their ordering here. This is a state where the activity in different regions is phase locked, so it's globally coherent. There's high global coupling in the spontaneous activity over time. You see an increase in that under DMT. This is before it, DMT, and then after a significant increase. That's also at a trend level true of the limbic system, which is intriguing. But where it's not true, in fact, where the rule is the inverse, is the high-level modes, these frontoparietal transmodal networks or systems, the default mode network and these other frontoparietal systems. When you look at the prevalence of these states or modes, they drop under the DMT. And we've shown this with psilocybin and LSD as well. So that for me helps me understand findings like this, where you might think, oh, this is a pattern that becomes, in a sense, more prominent under the psychedelic. And that's not true. It's one of the sort of confounding effects of blobs on brains. Really, it's that this pattern, this gold pattern, is breaking down. Its boundaries are breaking down. It's becoming more coupled, you know, it's traversing its usual boundaries and becoming more coupled with the rest of the brain. So functional module itself, it's actually becoming less present under the drug. And it's only through doing these dynamic analyses that we can see that kind of thing. Now, some of you in the audience might be seeing this kind of thing and thinking, high-level systems, high-level networks, high-level aspects of psychology, of mind function. What about this phenomenon of ego dissolution under psychedelics? You hear of it in spiritual contexts, and you hear about it with psychedelics, and you hear about spirituality and psychedelics. You know, there's something here. And of course, we've had the same intuitions, and I see effects like this, these high-level systems associated with things like self-reflection, particularly high-level aspects of cognition or psychology or of mind, and they are breaking down under the psychedelic. So yes, we can do those mappings, and yes, we see relationships, but I think on a high level, it's easy, and I share that intuition that maybe the breakdown of these high level, the psychedelics is a correlate of these high level abstract experiences, such as ego dissolution. And historically, we have done these plots. Here we're looking at a particular circuit within a particular high-level frontoparietal system, the default mode network. And yes, we see a decrease in coupling within a particular circuit there, correlating quite strongly with ego dissolution. I tend to show this slide these days to encourage people not to be so default mode network centric about the neural correlates of ego dissolution. In a sense, I think that's a mistake that I can hold my hand up and you know, confess some culpability for. But over time now, I'm starting to, influenced by the data really, think of this more as a DMN plus phenomenon. It goes beyond just a default mode network and seems to be true of a pattern that is, in a sense, is default mode network plus. It incorporates other high-level frontoparietal systems. And I'm motivated by that view by looking at these patterns. Here we're looking at the distribution of the 2A receptor. It's not just in the default mode network. Yes, you can see these are classic default mode network regions, the PCC and medial prefrontal cortex, but also these temporal cortical regions and lateral prefrontal regions that aren't classically default mode regions. We can look at brain evolution and see that it's not just default mode network regions that have expanded in our species versus, in this case, the macaque monkey, where we have the most data, but you look at chimp data, it's a similar pattern. It's a rule that's more true of just high-level frontoparietal, and not just frontoparietal, temporal as well, but high-level transmodal cortex. And I'm influenced by things like Daniel Margulies' work, looking at the principal gradient of functional connectivity in the brain that nicely separates unimodal cortex from transmodal cortex. I've talked about expansion across species, but what about within species, the human, and looking at cortical expansion through a lifetime, and similar regions are implicated here, undergoing the greatest expansion, these high-level associative transmodal cortical regions. We can look at connection length and how the fibers are longer in this high-level transmodal pattern. There's greater inter-subject variability and functional connectivity, lighter myelination, if that correlates with greater plasticity, or put it another way, where myelination is stronger, tracts will be more fixed. We can look at semantic processing and more abstract information processing in this high-level transmodal system. At about this stage, I usually start referring to it as the human system, with good reason. I just think it articulates what we're talking about here. We can look at temporal processing, longer temporal windows being processed by this human system, a certain type of metabolism, aerobic glycolysis, a particular faster type of metabolism associated with this human system, and psychedelics appearing to target this human system and breaking it down transiently, temporarily under the drug effects. So it all kind of ties in. And for me, it helps to understand what these drugs are doing to the mind and the brain, and help us understand the drugs themselves and their action, but probably more fundamentally understand the brain. A model, a guiding model for me in terms of thinking about the action of psychedelics and their therapeutic action has been this model that I introduced with Carl Friston a couple of years ago. We call it Rebus. Phonetically, it works, relaxed beliefs under psychedelics, that takes inspiration from hierarchical predictive coding, dominant model in cognitive neuroscience and increasingly in psychiatry as well. The essence of the model is to hypothesize where the principal action of psychedelics is in terms of hierarchical predictive processing. And it took me a while to work this out. I think in hindsight, Carl had told me years ago where to look, and I was like, yeah. But finally, it sort of sunk in that it was, and I do think this will hold up, the precision weighting of the models, of the assumptions, of the priors, to use the Bayesian language, the weighting of these models is what's affected by psychedelics. What happens to that weighting? It's relaxed. There's a lightening of the weighting. How can we think of precision weighting? Well, I guess the clue's in the name: precision, confidence. Our confidence in our assumptions is relaxing. Our confidence in our assumptions, in our beliefs, is lightening under psychedelics. And as you've seen these kind of things a million times, how you can read text like this, even though the details are jumbled up, because of our ability to see the world through our mental models. Here in the perceptual domain, you see something like this. Some of you might have seen this a few times. I certainly have. Not quite working from this angle, but you might hallucinate some motion, because something like this in the world would typically have motion. So you see it through your internal models. In a sense, you're hallucinating your experience of the world, because we see the world through our mental models. And so a natural next question is, well, how is this function encoded in brain activity? And traveling waves, cortical traveling waves, is a particularly juicy candidate. Some recent modeling and empirical work has backed up this assumption that cortical traveling waves do indeed encode these predictive processing mechanisms. With very simple experimental conditions of eyes opening and eyes closed, you can look at the direction of cortical traveling waves and see that with eyes open, the direction is bottom up. There's photic information to process, so it travels from the back to the front, from the back to the front. It's bottom up, and then with eyes closed, the directionality flips. It's top down, nothing to see here. And that's the picture with eyes opening and eyes closed. Now, our hypothesis coming in, looking at cortical traveling waves with a psychedelic, in this case DMT, we give the DMT intravenously, was that we would see flipping in the directionality of cortical traveling waves in a very similar way to what you see when someone simply opens their eyes. There's stuff to see, there's information to process, it now travels bottom up. So that's what we predicted, and that's what we saw in a very clear way. Here's the usual pattern with eyes closed, eyes opening, the bottom up flip, and the top down drop. And here with DMT, people have their eyes closed. We administer the DMT as a bolus. It's very rapid acting. People start feeling it in a matter of seconds. And they start feeling very, very immersed in an entirely other world that is visually rich, immersive. To call it dreamlike doesn't quite do justice to it. They'll say things like, that world felt as real as this world, if not somehow even more vivid. Very, very profound experiences under DMT. And here we see that flipping in the directionality of the traveling waves from top down to bottom up. Now, that was I think, quite a nice empirical demonstration or test of the hypothesis, but you can think of some of the intuitions around the model. If it's ego dissolution, if ego self is a particularly high level internal model belief, perhaps central in a sense, then to see its precision weighting or to intuit its precision weighting dropping down under psychedelics and how that may well map to ego dissolution or ego disintegration, and then different brain correlates of that phenomenon. You know, it all sort of ties in. And then if that's a thing going away, a lightening of the precision weighting of high level priors or beliefs, then what replaces it, if something relaxes, is there something that can come up, that can percolate up? And so this is just speaking to some classic phenomenology of the psychedelic experience, that these visions, in a sense, eyes closed visions, are very much part of the picture. Also, the release of emotion and memory is another part of the picture, and very much part of the therapeutic picture in terms of the therapeutic mechanism of action. I usually watch time, and I just said, yeah, okay. And so let's end with a very brief flight through this double-blind randomized control trial that we recently completed, comparing psilocybin therapy with an established antidepressant treatment, 6 weeks of an SSRI, escitalopram. I'm not doing justice to the design there, but if you want to ask me about the details of it afterwards, please do. The main findings were that the side effect profile or tolerability profile was pretty even across the two interventions, with some domains favoring psilocybin, less risk of anxiety or dry mouth in the psilocybin condition. It was 2 treatment sessions with 25 milligrams of big dose of psilocybin. The population is major depressive disorder, so depression broadly defined of at least moderate severity. Now, these are the efficacy outcomes. These are forest plots where we've given the confidence intervals here. If they cross 0, and all these bars are crossing 0, we feel at least, well, greater than 95% confident of a between condition difference. All these green ones are favoring psilocybin. Oh, what's this? It's the primary outcome measure. This was the anomaly in the efficacy measure mix, and because it was our primary outcome measure, it had to define the result. So if you read the abstract of this paper, you'll see it said there's no significant difference between these 2 conditions. I would encourage a bit more of effort in terms of looking at the actual data from these trials, because whether that is a true negative is, I think, fairly up for question. These are very, validated depression rating scales. In hindsight, I don't think we would have chose the QIDS again as the primary outcome measure, but HAM-D, Madras, BDI, all favoring psilocybin, less avoidance, more acceptance under psilocybin, anhedonia symptoms improving more with psilocybin, work and social functioning, flourishing, a domain of well-being, both of those, greater improvements with psilocybin, anxiety, greater improvements with psilocybin and suicidality as well. So it was promising. It's promising results, I think, in favor of psilocybin versus a gold standard treatment for depression. Also in terms of the speed of onset, faster improvements with the psilocybin. Now, this is important. A letter came into the journal, a few letters came in, and one of them said, discontinuation effects. Did you split the sample by people who weren't on any meds when they came into your trial? Presumably, there were quite a few of those, and those who were on an SSRI, an SNRI, or maybe some other psychiatric meds. And we said, well, that's an important question. You're right, we should look at that. So we split the sample up, and these are the findings. If you look at the majority of the sample, which were people who actually weren't on any medication coming into the trial, about 2/3 of the sample, then all of the efficacy outcomes, including that primary outcome measure, will significantly favor psilocybin. And the write-up of the paper, the whole narrative, would have been really quite different. It would have been, positive result here, psilocybin superiority versus escitalopram. However, if you look at it another way, the 1/3 or so who did come off an SSRI or an SNRI or any psychiatric medication, then none of the outcome measures would have favored psilocybin. Discontinuing your meds seemed to be a positive thing for going on to escitalopram, going back onto a med, and a negative thing for receiving psilocybin. It seemed to attenuate the therapeutic response to psilocybin. It's suggesting could be a psychological effect, sure, but it could be some residual neurobiological effect, maybe a downregulation of the 2A receptor, maybe that in the brain, plus peripheral things, meaning that going back on a med that your brain and body knows and only recently came off is going to feel okay. So fascinating difference there. And if we looked at the data in another way, plotting the temporal profile, these are those who didn't discontinue. This is psilocybin, all of these domains highly significantly favoring psilocybin. And this completely different picture if you look at those who did withdraw from their meds. The washout wasn't long enough. That's one obvious take home. It should have been longer than the 2 weeks that it was. But very important information, I think, going forward. So most of this work was done with my Imperial team, so thanks to them, and a big nod to Neuroscape, where I am now at UCSF. Thank you very much for your attention.
Thank you very much, Robin. This is the time to take a few questions for those who didn't ask any questions, either directly related to the material that Robin presented there that is associated to this topic.
Thank you, Robin, for your excellent talk. I practice in psychiatry and in addiction medicine, and I find all these evidence to be very exciting. My question for you is, how do we convince psychiatrists and addiction specialists about this new science and to think out of the box? I mean, there's a lot of evidence right now. It's just that I deal with a lot with refractory depression. And a lot of patients ask me about psilocybin, and then they're going to esketamine, which is now FDA approved. So how do we convince psychiatrists and addiction specialists about doing more research and actually move forward beyond our antidepressants?
Yeah, well, we present the data and we try to do it in a balanced way. And in a sense, it's not about effortful convincing rather than just this is what we're seeing. And my interpretation of it is that it's very promising. They're small trials, but there are bigger trials underway. I mean, the positive is that the work is happening. So if we're thinking of psilocybin therapy for depression, there's two organizations. One is a company, the other is a not-for-profit. Compass Pathways is a company, USONA, the not-for-profit, that are doing the required licensing trials. Compass are doing a phase 2B dose finding study that will segue into the phase 3 work. They're publishing or they're releasing the phase 2b results this year. And then that should lead on to the phase 3. And I think they'll run their trials in parallel, I think. And so, you know, milestones of something like 2024 when they would get readout on the phase 3. So it's happening. And you know, big figures in academic psychiatry are part of the advisory board around Compass, you know, former presidents of EMA like Guy Goodwin and APA with Paul Summergrad and others, you know, so very reputable individuals are sort of backing this work. And so I think it's just a matter of time and presenting the data. I think it's an equal consideration to manage expectation, actually. And because a lot of this is really exciting and it's easy to get maybe a bit carried away with it. So holding some of that in check and wondering about the pitfalls is probably an equal consideration of equal importance at this moment. So.
Other questions?
I have a question about, you had mentioned ketamine earlier, and with regards to ketamine, it being prescribed now or FDA approved and now being prescribed for treatment resistant depression, do you believe that not accompanying it with the clinical, you know, inpatient experience that you get with a guide, do you think that's going to have an effect on the therapeutic outcome of these patients?
I think it probably does. I mean, we can make it an empirical question, and people are doing that. So there's ketamine-assisted psychotherapy, CAP, being done now. And some of that work changes the mode of administration. It might be IM rather than IV. Even oral lozenge is being looked at with the psychotherapy. And so, you know, that's really taking inspiration from classic psychedelic therapy and making that that intuition, that inference that maybe we could get more, you know, bang for our buck if we add in psychotherapy here. And I think that's a fair intuition. It's just that the marriage is more obvious with classic psychedelics because the experience is it's deeper psychologically. It's more effectively intense. Ketamine is more of a dissociative than a psychedelic. Is it psyche revealing? I don't know. That descriptor, you know, however abstract it is, does work for psychedelics. You can understand, you know, in terms of the phenomenology, why it works. I think, you know, at least half of the participants in our depression research, people will be crying during the session, stirred by the music. You don't really see that kind of thing with ketamine so much. So there are differences. So potentially, yes, but also maybe the subjective effects are different with ketamine. And maybe, you know, let's not be too biased about psychotherapy. Maybe there's some limitations with psychotherapy. It's expensive. It would be efficient if you could get away with just a drug. In fact, you know, that's dominated psychiatry for many decades now, the efficiency of drug therapy, you know, pharmacotherapy. It's convenient, it's cheap, but is it, you know, can we do better with combination treatments? Can we get a deeper kind of therapeutic action?
I think one of the hard problems of consciousness is trying to find the neural correlates of it. And, you know, there's some really exciting evidence as far as the serotonin 2A receptors and location of it. Do you think that the use of psychedelics could be used to find the neural correlates of consciousness as far as ego dissolution goes and stuff like that?
Yes, I do. And that way that we've referred to this for quite a while is, in my view, the right way. And it brings us into the domain of philosophy. You know, we're talking about neural correlates, which is basically saying that the experience of phenomenology is mapped by what we're seeing in terms of the dynamic neurophysiology. And so we can look at one side of the coin, which is the experience itself, and then we look at the other side, which is the mapping, the correlates. But whether there is a bridge that's bridgeable there in any other way than making mappings is actually something that I often doubt. It just may be that when we look at the biological correlates of these experiences, there's something about their dynamics that sort of shouts the phenomenology. And I think that's where we'll come to eventually, where you almost get a sense for what it feels like by looking at the biology. Yeah. And there it will be the level of mapping that's really important, that you don't go too low. But something at you know, a whole brain level, not just brain, but body, brain-body coupling, might say, this is it, you could see it, you know, and it's compelling. Yeah.
Sorry, I just had a question on the potential link between psychedelics and religion. Possibly, I know that because of Brian Murarescu's book, The Immortality Key, they're conducting a study at Harvard for, I think, the link with ancient Greece. But I was just wondering, maybe something with like Christianity or Judaism, or yeah, if you have any information.
Yeah, so I had to, you know, I shipped over a lot of books, and it might be somewhere in the middle of the ocean right now. And one of them is Brian Murarescu's new book that I'm really excited to read, and I have to wait until it arrives. It might be 2 months away, so that's one that's kind of bugging me. I ship too many books, so they're, you know, they're heavy things. So I haven't read it, but I think the essence of the sort of thesis is that psychedelic experiences lie at the origin of the birth of religion, I think. And was Brian the first to say that? I think he would admit that he's not. But maybe he's said it in the most complete and up-to-date, you know, well-referenced way. And so I can't say anything more informed than that until I've read it. But Brian Murarescu, what's it called, the Immortality Key? Yeah, it's going to be an interesting one. And you know, Terence McKenna wrote about the birth of human consciousness and his hypothesis that use of magic mushrooms by early proto-humans led to the, you know, catalyzed the evolution of the human brain and human consciousness. That's an interesting one. I don't buy it, but now there's this recent data around the 2A receptor and cortical expansion that puts a new modern spin on that idea that might be saying it wasn't magic mushrooms, the ingestion of magic mushrooms that did it, but maybe there's something about serotonin, conditions like adversity, think of evolutionary pressure, you know, human beings' adaptability in a broad range of environments. Why is that? It's quite exciting, you know, to think that maybe adversity and our ability to adapt lies at the heart of why our cortices expanded as they did. The thing with evolutionary solutions like that, you get a sense that it's never just one thing, you know, it's a combination of things, but that's an interesting one. Yeah.
One last question. Go ahead.
Yeah, so my last question is regarding plasticity. In the studies that you mentioned earlier, within the demographic of your guys' participants, did you find an age-dependent difference in plasticity changes?
We haven't done that much in humans looking at markers of plasticity. A lot of that work is lower level, you know, rodent work or in vitro work. And so we do have some functional readouts of plasticity. which through back translation correlate with actual anatomical plasticity. We have this visual LTP paradigm, long-term potentiation, and we have got some unpublished data finding enhancements in that marker, that proxy, if you want, of neuroplasticity with psilocybin, and an age-related effect. So that there are, age-related differences in 2A availability. It seems to sort of, well, it's high in the young brain and it drops away. And I think there's quite a steep drop into middle age. Whether that relates to anything like how our sense of assuredness tends to come into play around middle age, I don't know, that's loose, isn't it? But maybe. I mean, Michael Pollan in his book writes about psychedelic experiences for, in a sense, the hardening of belief structures in more mature individuals, sort of in the latter periods of one's life, which is interesting. It's not necessarily talking about pathology in aging or end of life kind of periods, but more, psychedelics for midlife crises or something, I don't know. But we need to look at it as the obvious thing. And we haven't yet found anything compelling, but we haven't looked well enough, I think, yet. So maybe big samples are needed to look there.
Well, thank you very much, Robin, again. And I just want to tell you that in my opinion, psychedelics mark some kind of a revolution in psychiatry and in certainly clinical psychology, psychiatry, mental health treatment, and so on. And the work that Robin is doing and others, bringing psychedelic drugs into the fold of science is helping them some credibility with our understanding of consciousness, things that are related to brain mechanisms and so on. And I'm very excited about Robin being in California and Thanks again.
Thank you, Amir. Thanks, everyone.